like always. And Ana María Gómez is our speaker, and it's really, I'm really delighted to have her as a speaker today. So Ana received her PhD from the Complutense University of Madrid, where she started her long and productive study of cardiac hypertrophy and arrhythmias. In fact, the connection between hypertrophy and arrhythmia is something that Ana has taught us over the years. She then traveled to the US to John Lederer Lab, where she did really pioneering work on sparks, describing calcium sparks in normal and hypertrophied hearts. This work resulted in a beautiful, really beautiful and important paper in Science in 1997. From Baltimore, Ana returned to Montpellier in France. And Montpellier, I can attest to, has beautiful beaches and one of the oldest medical schools in Europe. At present, Ana directs the signaling and cardiac pathophysiology unit at Inserm in Paris. She is also the present chair of the European Society of Cardiology Working Group on Cardiac Cellular Electrophysiology. And Ana will tell us about a new mechanism in CPVT. So Ana, thank you again for joining us. And the screen is yours.
Okay, so thank you very much, Joram, for the invitation and for this nice introduction. So I will start
one second.
Yes.
I forgot to say one important thing. We cannot interrupt Ana in the middle because we have over 130 people that are attending this. And so it'll be very interrupting if we do. So please put your questions to all in the chat and I will read them at the conclusion of her talk. Thank you, sorry.
Okay, so Catecholaminergic polymorphic ventricular tachycardia, or CPVT, it's a genetic disease, as I think most of you know, that it's manifest by syncope or sudden death, mostly in children or young adults under conditions of physical stress or emotion. Most of the mutations that are identified up to date, they are related to the Ryanodine receptor type 2. Ryanodine receptor type 2 is a calcium release channel. So here you have is an homotetramer. So here you see only one monomer. And each of these dots is some of the mutations that have been identified in CPVT up to date. There are more than 150. And they are clustered in 3 regions of the channel, in the C-terminal region, in the central, and in the N-terminal region. The mutation that I will present to you is in the N-terminal region. So some other mutations are not in the Ryanodine receptor, but are in proteins that binds to and modulate Ryanodine receptor function. So these proteins are calsequestrin, that is a calcium chelator protein inside of the sarcoplasmic reticulum. Triadine is a protein that links the Ryanodine receptor and calsequestrin, and also calmodulin, which it's here. So it's only one it's presented, it combines. Then there are also other proteins that have been related to the CPVT mechanism, not because they have mutations, but because some mutations of the Ryanodine receptor have been shown to interact with the binding of SKBP 12.6 and also on the phosphorylation states. So the Ryanodine receptor main function in the cardiac myocytes is to release calcium to activate contraction. So in each heartbeat, the open of the L-type calcium channels during the action potential lets some calcium enter into the cell. This calcium is not enough to activate contraction, but it activates the ryanodine receptor that amplifies this signal by releasing a much bigger amount of calcium to the cytosol. Then the cell relaxes when this calcium is pumped back to the sarcoplasmic reticulum by the Cerca pump and also extruded out of the cell through the sodium-calcium exchanger. So the sodium-calcium exchanger is electrogenic, and for each calcium that is extruded, there are 3 sodiums that goes into the cell. So it's one net positive charge. And it has been, this mechanism has been proposed like a physiological mechanism for the pacemaker cells. So it has been proposed that calcium release at the end of the slow depolarization phase may, by activating sodium-calcium exchanger, further depolarize the cell to bring to the threshold for the action potential. In ventricular cardiomyocytes, the main function, as I told you, is during systole to activate contraction, but in some conditions, pathological conditions, if there is calcium release, enough calcium release during diastole, it may, by the sodium-calcium exchanger, produce delayed after depolarizations. If this delayed after depolarizations of DADs reach threshold to activate one action potential, then we have a trigger activity and we may induce arrhythmia. It can be the trigger of the arrhythmia. So this is the mechanism that has been proposed in CPVT. And so I was contacted by a collaborator that is a cardiologist in Spain, Dr. Sorio, who had a family identified with CPVT, diagnosed with CPVT, where she identified this R420Q mutation. Because we have access to the patients and because this mutation is in the N-terminal portion where there was at that time not many studies, and not in mice at least, we decided to analyze this mutation and try to find the mechanism of this. So the first thing that we did is to create a knock-in mice. So these mice have the same mutation. Like that, we can have electrocardiograms from the mice, and we can have also isolated cardiomyocytes. In parallel, we got blood samples from 4 members of the family, 2 with the mutations and 2 without the mutations of either sex, to get induced pluripotent stem cells and differentiated into cardiomyocytes. So like that, we can have these cells that are human, also they are immature and they are automatic activity. And we can have also the mice that are mature, but they are mice. So the data that I will present to you today, it will be mostly taken from the mice, but some data that it's just, also I will present you from the iPS-derived cardiomyocytes. And when it's from this, is in the blue square. So just if I forget to tell you, it's inside of the blue. So just after doing the mice, the first thing we did is to verify that the mice have the CPVT phenotype. So in this CPVT, to find the phenotype, what we usually do is to inject epinephrine plus caffeine, and the mice show this bidirectional ventricular tachycardia. And all the mice that of the knock-in mice that we test have this phenotype and none of the wild type. So we validated the model. With these electrocardiograms, we led during 24 hours to analyze the electrocardiogram of each mice. And we found that during the daytime, so during rest, there is, well, there was not significant difference in the heart rate in males, but there was slower heart rate in female mice. So this suggests us that it may be some difference in the pacemaker. And as all cardiomyocytes, they express Ryanodine receptor type 2. So we went to see what happened with this family, with the heart rate before beta blockers, or at the beginning of the study. And there was someone that were bradycardic, but if we took all data together, there was the females, the women who have also smaller heart rate. Looking more at the electrocardiograms, we found some junctional escapes both in mice and in people, and so we say that this mutation is accompanied by sinoatrial node dysfunction. So we started this study by analyzing the pacemaker cells from the sinoatrial node of these mice. So just to remember what is the mechanism of action potential generation, because the pacemaker cells, they are automatic, so they generate action potentials. And this is because during the diastolic period, there is a diastolic depolarization and different ionic channels are involved. So the IF is the main, the known pacemaker current. So this is involved in this depolarization phase. The T-type calcium channel may be also involved. It has been proposed that calcium release in the cytosol from the ryanodine receptor can also be involved by activating the sodium calcium exchanger. So this release, it could be, we don't know if it can be spontaneous completely, if it can be synchronized by T-type. We saw a few years ago that the CAV 1.3 induces, it triggers some calcium release at the end of the depolarization phase. And this, what it does is through the sodium calcium exchanger, depolarize the cell, arriving to the threshold to activate the CAV 1.2 and the action potential. And then we have a calcium transient. So to analyze this, what we set up a preparation of the mice sinoatrial nodes, you can see here. So we saw at this part, and we loaded with calcium dyes and look at it in the confocal microscopy, so we can see the spontaneous calcium transients in individual cells, but within the tissue. We didn't isolate the cells, and maybe you can also see in the video some calcium sparks during the depolarization phase. So then we did, like with isolated cells, we did one line scan and analyze the calcium over the time. So we have a resonant microscope, so we can have good time resolution. And here, the first thing that you can see here, this is a wild type and a knock-in sinoatrial node cell. And if we analyze the time between two consecutive calcium transients, this is the cycle length, and we saw that this was prolonged in the knock-in mice. So this goes to the slower heart rhythm that we saw in vivo. In this preparation, we didn't see a difference between preparation from males or from females, so they were all prolonged. Then we wanted to analyze the voltage, the name voltage clock. So we did patch clamp, we isolated the cells and did patch clamp to analyze IF. And here you see in the squares, the gray squares are the knock-in, and we didn't saw any difference in IF. When we analyzed the calcium current, the L-type calcium current, we didn't saw a difference in the amplitude of the calcium current between wild type and knock-in. And this is in the patch pipette, we had BAPTA to chelate the calcium. So if we don't chelate the calcium, we repeated the experiments with no chelation. In that case, we see a decrease in the current density. So this decrease in the current density may be involved in this prolongation of the cycle length. So this slower of the rate. but it seems that it's dependent on the intracellular calcium. So if we go back to look at the calcium signal, so this is, you can see here the calcium sparks or late diastolic calcium release before the transient, we could see that we have more in the knock-in. So we think that maybe this could be inactivating by the calcium-induced inactivation, the L-type calcium channel. So we also analyzed, because the mutation is in the Ryanodine receptor, we wanted to analyze the characteristics, or the frequency and characteristics of calcium sparks. So we analyzed this during the diastolic periods, and we found, so the time is extended, as you can see. So if we count this calcium release, we can see that the frequency of the calcium release is higher in the knock-in, so we have more calcium sparks. And if what is also striking is if we see the duration, these calcium sparks were much prolonged. So of course, if we calculate like the calcium release, some calcium release through calcium sparks, so we can multiply the mass, so that the mass takes into account the duration, the amplitude, and the width of the calcium sparks. And by the frequency, we see that there is more calcium leak during diastolic periods in the knock-in cells, knock-in mouse. So for this pacemaker study, what we found is that we have longer calcium sparks. So the conclusion is that these prolonged calcium sparks and bigger, it should be taken into account, like it enhances calcium clock, but in fact, what it produces, it inactivates the calcium channel. So in fact, it depresses the voltage clock, if we can name it here. I know Dario is here, so maybe he doesn't agree. And then this is involved in bradycardia. So then, of course, this is interesting because when there is stress and ventricular cells became more active, if the sinus rhythm is slower, it may be more prone to be arrhythmogenic. But what we wanted to really understand is what happened in the ventricular cells. So we went back to the mice and analyzed the electrocardiograms. So this is the same than before with epinephrine plus caffeine, just to show that the mice have this phenotype. And then we tried two other kinds of stress. So this is accepted to be, or proposed to be, like emotional stress, that is blowing hair with a hair dryer during 10 seconds, 10 times, 1 time a minute. And then we can see that this procedure produces ventricular tachycardia. So if we count the ventricular tachycardia episodes in this protocol, we see that there are some also in the wild type, but in the knock-in, there are much more episodes of ventricular tachycardia, and they were prolonged. If we injected isoproterenol, we could see also here, you see 3 ventricular ectopies, and we saw that under isoproterenol, we have also more episodes in the knock-in. So if we measure with this ventricular tachycardia, they also show it in the mice. So then we isolate the ventricular myocytes to analyze the electrical characteristics of these cells. So we did patch clamp and did patch clamp without calcium chelator. So here, these 4 examples are with isoproterenol. So in the wild type, these are the action potentials that we found. And in the knock-in, we saw some DADs, as you can see here, and also some trigger activity. As you see in the summary data, we have more proarrhythmic events in the knock-in. all this was completely blunt if we repeated the experiments chelating the calcium with VAPTA. So all this is related to intracellular calcium. We did microelectrodes in the iPS-derived cardiomyocytes. So this is the action potentials in this kind of cells. So these are humans. And this is with microelectrodes. It's not with patch clamp. And we also saw under isoproterenol DADs in these cells that came from the mutant carrier. And we saw the mutant carrier in basal conditions, some of them, and this was much higher with isoproterenol. So we found DADs in both mice and human cardiomyocytes. So these are the electrical signals. Because the mutation is in the Ryanodine receptor, we went to analyze the calcium, intracellular calcium handling by confocal microscopy. So here we give this protocol, a train of electrical stimulations at 4 Hz, and then stop and see if there are some activity. So in control conditions and with isoproterenol. Isoproterenol induces some calcium waves also in the wild type. In the knock-in, we found even in basal conditions some cells presenting calcium waves. Only one, okay. And with isoproterenol, we have half of the cells presented in this behavior with calcium waves after the train. This protocol cannot be do exactly the same in human-derived cardiomyocytes because they are automatic, so we cannot stop and see. But we stimulated these cells at 1 Hz that you can see here. This is a mutant in basal conditions and with isoproterenol. These are the electrical stimulations. And we could see in diastolic periods also some like big calcium spars or small calcium waves. And this was seen also in basal conditions and increase with isoproterenol. So far we see that these cells have this proarrhythmic behavior in the electrical activity, and this is accompanied by calcium waves. So what happened with the calcium transient? So because we have them so to analyze, because normally this person have normal function. And so here we analyze the calcium transients at 2 Hz and at 4 Hz, just to see if there is some difference that it's frequency dependent. So in red is the knock-in and in black is the wild type. And we didn't see any difference in the calcium transient amplitude at 2 Hz or at 4 Hz. And in the K time, it was also similar at both frequencies between knock-in and wild type. When we added isoproterenol, we have this increase in the calcium transient amplitude in both genotypes, but this increase was a bit small in the knock-in, and the acceleration of the decay time was similar in both groups. Because we added caffeine, because to estimate the amount of calcium into in the sarcoplasmic reticulum, we measure also this caffeine evoked calcium transient. So this is like one index of the calcium content. The same in black, it's with it's a wild type at here knock-in. Here it's only presented at 2 Hz, I think. And we see that there is a decrease in this amount of calcium stored in the knock-in. The kinetics were similar. And isoproterenol increases the load in both cells, to the same level. So maybe there is, so what we found, the difference that we found here is that there is a decrease in the SR calcium load, because we didn't find a difference in the calcium transient amplitude, but we saw a difference in the SR calcium load. we thought that maybe the fractional release is changed. So the fractional release, we calculated like how much calcium is released by a given amount of calcium. So in each cell, we normalize the amplitude of the calcium transient, but by the amplitude of the caffeine-evoked calcium transient. And this was higher in the knock-in. So by a given amount of calcium stored in the sarcoplasmic reticulum, there was more calcium release that at the end became normal calcium transient. And this was increased by isoproteinol. So this is in mice cells. Is it just dependent on the species? So we did this kind of analysis in human-derived cardiomyocytes from the same mutation. So these are the calcium transients, very weak, but okay. And then we had caffeine. So this is the same cell before and after, the same cell before and after. And when we calculate the fractional release, we found, like in the mice, that the mutant show higher calcium release. So just to summarize the data that we have so far, we found that this mutation is associated with VADs and calcium waves under isoproteinol. They have normal calcium transient. They have lower SR calcium load, and they have higher fractional release. So this is common to many other CPVT models. And we wanted to know why we see these things. So in some years ago, we found with this other mutation that is in the C-terminal portion of the channel, we saw that this particular mutation have higher calcium sensitivity. So what we did is to permeabilize the cells and bath the cells with this different concentration of calcium in the cytoplasmic part, and count how many calcium spikes there were on each cytosolic calcium in wild type and in the mutant. So we wanted to do the same. We did the same kind of analysis with this N-terminal mutation. And this is the data at different of calcium spark frequency at different calcium concentrations. And there was not very striking difference. So maybe there was more at higher calcium, but it was within the limits of the significance. So because calcium sparks, when we have a lot of calcium, they came together and we cannot really count them apart. We did binding assays. So as ryanodine only binds to the channel that it's open, if we analyze the binding at different calcium concentrations, we can see also the calcium dependence, and it was not significantly different. So it doesn't seem that at least the cytosolic calcium sensitivity changed. Then we measured similar things in taking into account the calcium inside the sarcoplasmic reticulum, because the fractional release is bigger. So by the same amount of calcium stored, we have more calcium release. So these experiments are taken also in permeabilized cells. And there are two different kind of dyes. So this is the cytosolic signal, so we can see the waves. And this is the signal inside the sarcoplasmic reticulum. And we can see for each wave, we have a depletion. And then with caffeine, we depleted. So we calculated the threshold of the calcium wave with the amount of calcium stored, and this was decreased. So it's easier to have calcium waves by the same amount of calcium. So because this data is in quiescent cells, we wanted to analyze calcium-induced calcium release function of the channel. So we did simultaneously patch clamp and confocal microscopy to analyze the calcium-induced calcium release and the gain. So this is an example in the wild type at 0 millivolts, and this is in knock-in. So the same in black is the wild type. So if we just look at the L-type calcium channel, the current density at any voltage, it was not significantly different. So we found normal calcium current, but the calcium transient that was triggered by this calcium current was bigger in most of the voltages that we analyzed. So if we calculate like the gain of this calcium-induced calcium release, so how much calcium is released by the integral of the channel, this is at -20 millivolts, something like that. We see that this is increased. So also for this channel is more active by a given amount of calcium in the sarcoplasmic reticulum, but also by a given amount of calcium entry. So maybe that is why even if the load is decreased, we can have normal calcium transients. So because in one of the old papers that Joram just mentioned, we found changes in gain of the calcium-induced calcium release in heart failure when there are some, there is some difference in the T-tubule structure. We wanted to analyze at this junctional sarcoplasmic reticulum and T-tubule. So we did electron microscopy of these cells, of these hearts. So here you have one example. The first two are color. So you see in yellow is the intraluminal sarcoplasmic reticulum. In green is the opening of the T-tubule. So this is extracellular membrane. And these dark, more dense spots are the feet of the ryanodine receptors. And so these are some examples in wild type and in knock-in. And what we saw is that some of these couplons of the knock-in, they have like, if we measure the length of the couplon, they were shorter, as you can see here, compared to there. And the junctional sarcoplasmic reticulum was a bit wider in some cases. So there was also some that looks more normal, but some that looks very much widened. So we wanted to see what happened in the calcium release units. If we look with a super resolution microscopy, so we use a gated step, and you can see here the clusters of ryanodine receptors in the wild type that are well aligned, and in the knock-in there was a bit less aligned, and an average was a bit smaller. So this is the first time in the mutation of the ryanodine receptor that there are this kind of ultrastructural disorganization or alteration. So we were very striking of why a mutation, a point mutation in the ryanodine receptor can have this structural effect. So because the calcium sparks originate here, we wanted to analyze the function of this calcium release unit. So we analyzed the calcium sparks. And as in most of the CPVT models, we see more calcium sparks in the knock-in. So you have here 1 and 2, so you can have here. And when we add isoproteinol, we have more in each case. So I didn't show here the data with isoproteinol, but it was the higher calcium spark frequency was in the knock-in with isoproteinol. As we saw that there was longer calcium sparks in the pacemaker cells, we also, that have different structure, we also analyzed here the characteristics of calcium sparks. And we found that these calcium sparks in ventricular cells were also prolonged. So you can see here this baseline and baseline wild type knock-in. This was prolonged. And this was even more prolonged with isoproteinol, beta-adrenergic stimulation. So then we see, okay, so why this, of course, there is a mutation, but we found this widening of the junctional sarcoplasmic reticulum. And we know that the spark termination, one of the mechanisms that has been proposed is the local depletion. So we think maybe if the junctional sarcoplasmic reticulum is wider, the local depletion is a bit slower. So we try to mimic some swelling of the sarcoplasmic reticulum by permeabilizing the cardiomyocytes and then bathing them with hypoosmotic solution. So these are all wild type, they are not knock-in. This is the frequency of calcium sparks in this normo-osmotic solution and hypoosmotic. And the frequency did not change by this maneuver. We look at the duration and we didn't find a significant prolongation. It depends on the statistical test, but the correct test that if we didn't do a t-test is significant, but we need to do another kind of test because it's not normal distribution and it's not significant. So we cannot conclude that the change in the widening of the SR is prolonging the calcium sparks. It may contribute because we see a tendency, but maybe not enough or maybe nothing. And of course, it cannot explain the increased frequency because it didn't change the frequency. So we think that there is something else in the channel that it's doing, that is changing maybe the gating. So we were also collaborating with Spiros Tissinopoulos, and so if here is also the ryanodine receptor, only 2 homomers are here, and the N-terminal portion is in dark blue, and some other mutation in the C-terminal has been shown that it may be some intramolecular interaction difference. So we wanted to see if this mutation that is found in this region may interfere with intramolecular interaction or intersubunit. We didn't see any change in the intersubunit. That was the first hypothesis. But we found that there is a tighter interaction between the N-terminal and the C-core solenoid in the mutated. So maybe this may interact with the closing of the opening of the channel. So why it doesn't work? Okay, so the next thing we did is to analyze the single channel current. So we did that in collaboration with Josefina Ramos Franco. And so this is a channel. You can see the close in bottom and the open in top at in this example, 2 different calcium concentration. And you can see that the channel is open and closed in this calcium. And with higher calcium, the probability of being closed is smaller and the probability of being open is bigger. When we do the same analysis in the knock-in, we found that in between the close and open, there is a big probability of being open in subconductance. So you can have different subconductance here. So for example, at this, that makes, if you compare at low calcium, that the channel is more open than the wild type. But even if it's at subconductance state, and also they are longer. And this appearance of subconductance depends on calcium. So they are much higher when there is low calcium. So this may, even if it's in low conductance, it may produce calcium leak. So we analyze the calcium leak with this Shannon protocol. You can see here on better because here you cannot see much. So we analyze the diastolic calcium leak total, because with calcium sparks it's only on a spark visible, and this was bigger. So we found increased calcium leak. So if we summarize up to here, we found higher fractional release, higher calcium-induced calcium release gain, and more frequent and prolonged calcium sparks. if we want to know why the mechanism of these more frequent and prolonged calcium spikes, we saw for the prolongation, maybe it's because there is altered intramolecular interaction and also longer openings in subconductance. And all this increases sarcoplasmic reticulum calcium leak. But there may be other possibilities. It can be changes in the phosphorylation, or it can be changing in the accessory proteins. So we analyze these other possibilities. So we start by phosphorylation because some of the mutations in the C-terminal that has been shown like it is more phosphorylated. We also try FKBP because the same authors have found that the CPVT, some of the CPVT mutations that they analyze are less bound to FKBP 12.6. And then we analyze the other proteins because as mutations in those proteins are involved in CPVT, if the mutation in R420Q interferes with binding of these proteins, like calsequestrin, triadine, and calmodulin, it could also be one of the mechanisms underlying the frequency. So we started by the phosphorylation. So first of all, the global expression is similar, Ryanodine receptor. So the mutation does not alter the level of the expression. And we analyzed the phosphorylation in the equivalent sites of 2808 and 2814. And we didn't find a difference in the phosphorylation status in these sites between wild type and knock-in. So it doesn't seem that the phosphorylation is involved. We also try a new site that has been described lately, that is the 2367, that is an inhibitory site instead of an activatory. And this was similar. And the spec, that is the protein that phosphorylates in this site, was also similar. So we didn't find any difference in the phosphorylation. Then we went to look, as I told you before, the FKBP 12 or 12.6 protein. So with these are binding, these are co-immunoprecipitation experiments. And so we found that the binding of the FKBP 12 or 12.6 to the ryanodine receptor was similar between knock-in and wild type. Because this protein has been also pointed to be involved in this CPVT, we wanted to check if in human cells it's different. And so we repeated these co-IP experiments in human-derived cardiomyocytes, and we didn't find a difference. So this mutation does not alter the binding to FKBP 12.6. What happened with the other proteins? We didn't find either any difference in the binding of calmodulin, calsequestrin, or triadine to the ryanodine receptor. So then we have more frequent calcium spikes. We have no alteration in expression of phosphorylation or accessory proteins involved in CPVT. But we have a nanostructural alteration on the calcium release units. So this was still striking. So why a point mutation can alter the structure of the dyads. So then we remember of junctufilin 2. So junctufilin 2 is a protein that it can bind both to ryanodine receptor and LTAC calcium channel. It holds the sarcoplasmic reticulum junctional close to the T-tubule structure. And moreover, it has been shown that when it's overexpressed or down expressed, it modifies the frequency of calcium sparks. So we say it's a good candidate. So we look at junctufilin binding to this mutated ryanodine receptor. And as you can see here, there was much impaired, so it was very much striking. So there is a decrease, significant, you can see here, significant decrease of the juntufilin binding to ryanodine receptor. And however, the abundance of juntufilin was normal, and the binding to L-type calcium channel was not modified. So this result we wanted to verify in human-induced cardiomyocytes. So here you can see the binding to juntufilin. So these are 2 control samples and 2 samples from a mutant carrier. And we can see that the ryanodine receptor that was bound to juntufilin was significantly depressed in the mutant carrier. So if we take all data together, we can see that we have found more and longer calcium sparks, calcium waves, DADs, and arrhythmia. That is what we have found. So why we have this behavior? So the mechanism that we have found is when it's a intramolecular interaction, we have found a tighter association between the N-terminal domain and the core solenoid. And maybe this is involved in longer channel opening. What we have seen is that we have impaired binding to juntufilin 2. So if we have impaired binding to juntufilin 2, this, because of the functional role on the ryanodine receptor, may be involved in calcium sparks occurrence. So both more calcium sparks and longer calcium sparks, more and longer calcium sparks can be because of these 2 modifications. And also this impaired binding to juntufilin may induce a nanostructural alteration of the diet. So if this is a normal diet, you have 2 here in the wild type. And when this one makes a calcium spark, it just stays there. It doesn't propagate and it's not pro-arrhythmogenic. In this remodel mutation, we have wild type ryanodine receptor that may bind to juntufilin 2 normally, and some that are mutated and don't bind to the juntufilin. So we have shorter couplons and wider ryanodine receptor, wider sarcoplasmic reticulum junctional, and some may be because we saw that they are not all well aligned when we saw the super resolution. So in this setup, if this channel that is more prone to open, it may trigger the opening of this one that is nearby and propagate to the next one. So we think that this may be, we think that this may be the mechanism of this mutation. And so I just want to end by thanking, so this is my laboratory in Châtenay-Malabry. So now we are not in Montpellier, but we are here. And of course, this is the picture before. It was during COVID. This was before COVID, the calcium team. And those who have participated in this work, and the 3 first authors, Li Henjin, Alexandra Zaratnikova Jr., and Ricardo Risetto, and also of the collaborators that are very important for this work. So in Europe, in Montpellier, in France, Mangoni's laboratory, in Valencia, Thorio's laboratory or service of cardiology, in UK, Spiros Sissimopoulos, in Italy, Protasis lab, in Switzerland, Niglis lab, and in United States, Valdivia's lab and Ramos Franco. And thank you for your attention. I can get questions if you want.
Well, that was a long journey and a beautiful one. Thank you, Anna, really, for describing so clearly a comprehensive and systematic set of experiments to explain the mechanism. So it's greatly appreciated. So let me ask you one question before I read questions from the chat. That's really about the early part of your talk about the pacemaker. And I think what you mentioned was the mechanism is through the calcium clock, that there is, more release, longer sparks causing inhibition or inactivation of calcium current, and therefore reduced calcium current, and as a result, a slower rate in bradycardia. What happens if you give ISO on top of this mutation? Is the rate increasing?
It increases, but increases less than in the wild type.
So even the dependence on beta-adrenergic is reduced.
Yeah, they are much more. I mean, the cells respond to the beta-adrenergic stimulation, so it's not that they are not responsive. We have much more calcium sparks or waves in the diastolic, but we have less acceleration than in the. So I don't think that it's so clear that there are two independent mechanisms or three, calcium clock, voltage clock, and they are not independent. That it's what I think, what I think from my data. And if you destabilize the equilibrium, I think I see there is, if you have more calcium, even if calcium is involved, if you have too many, you interfere with the voltage clock.
Yes.
And then you have.
So what happens in the patients? The phenotype is bradycardia at rest, and then arrhythmias or DADs.
Yeah, not all are bradycardic, but there is much more. Many patients are bradycardic.
Yeah. And then with exercise or emotional stress, they display the triggered activity arrhythmias.
Yeah.
Okay, let me get to the list of questions here and see what's happening. So, the first question is from Dario Di Francesco in Milano. And Dario is saying, very nice data, Anna, thank you. Have I got it correctly at the beginning of your talk that the R420Q knockout mouse is bradycardic? That would sound strange in the presence of more frequent and prolonged calcium sparks, unless other mechanisms are involved. But then also, it was was unchanged. That's the question from Dario.
Yeah, so I think some of these of his questions. So thank you, Dario. I think some of your questions I already answered. So the mouse is not bradycardia, I think, because of the autonomic tone. If we inject atropine and propranolol, yes, they are bradycardic. So of course there are other mechanisms. That is what I think I just was explaining to Joram. So if we think that this mechanism of more calcium sparks getting slower in the pacemaker, it's because we have more calcium inducing activation of the calcium current that it's also involved in the pacemaker activity. But IF, we didn't find a difference. I don't know if, yeah, no, we don't find.
So is there any evidence that IF is or is not sensitive to calcium? Is it, we can rule out the possibility that IF is calcium sensitive?
I let Dario to answer. Do you want to answer, Dario?
That's legitimate.
Yeah. What did say? Okay,
Dario,
now he's on mute. Okay.
There is no doubt, and Antonio is also in the group there, and I'm sure even if it's not obvious, he is smiling now. there's no doubt that calcium does not directly affect the fatty channel. On the other hand, there are several ways by which calcium can affect the channel, one of which is obviously through indirect phosphorylation mechanisms. So the situation is rather complex, but certainly there is no direct effect of calcium over.
Yeah, that's what.
But maybe if the cell is a bit depolarized, I don't know if the cell is a bit depolarized, you have less activation of the ions.
Sure.
Also.
Absolutely, yes. Absolutely, yes. That is an indirect.
It's also another indirect effect. That
Absolutely, yes. Yeah.
Okay.
We cannot see.
Okay. I mute myself. Thank you.
Yeah.
Okay. Thank you.
Okay. The next question is also from Milano, from Antonio Zaza. So Antonio asks two questions, I think. One of them is SR T-tubules disarray is typical of hypertrophic remodeling. Can you rule out the secondary nature of the structural changes? That's question number one.
Okay, so the question number one, in hypertrophic remodeling, as far as I know, at the beginning, so when it's more like compensatory, it may be more proliferation of T-tubules. When they are more heart failure, we have less T-tubules. But we don't see a difference in this kind of difference. So what we found, we have the same T-tubules. It's just I mean, we didn't really measure, but it doesn't seem to be difference in the T-tubule. The cells are not hypertrophied. The heart is not hypertrophied. So it's more at the couplons. And it has been shown in calsequestrin knockout, this kind of whitening of the junctional sarcoplasmic reticulum. That's why we measure calsequestrin, but we didn't find a difference in calsequestrin. But no, in hypertrophy, so these cells are not hypertrophied, and in hypertrophy, there is not the same, as far as I have seen, similar changes. No, I don't.
Okay.
I don't know if anybody has analyzed junctophilia in heart failure. I don't know.
But you don't see as much disarray in the T-tubules as you see in some other.
No.
Okay, yeah. Okay, the second question from Antonio here. The mutation is in the PP1 docking region of RYR. Did you look at PP1 binding?
No. No, I am not sure that it's the location of the PP1. So this mutation has been analyzed, crystallized by Philip van Petegem, and it's very hidden in the. I think I have. No, not this. Here. I think I have one, if I can, no, I cannot show. Yes, so it's here, the mutation. So this is the N-terminal that is in color, that is the domain A, B, and C, and the mutation, is in the center. It's, no, okay, I thought I have here, yes. No, you can, no, okay. So anyway, it's I don't think this site is on the PP1 binding, but I will check.
Well, I just I would say broadly, I'm not sure what is exact location of the PP1 binding site, but it's approximately in that region of the protein. So mine was just a guess.
I thought that the PP1 it was more on the central region, but I'm not sure. So
well, from
I will check.
I've never checked it myself, but from the reviews I read,
I will check. But there is no difference in the phosphorylation status. But we didn't look
true.
at PP.
But I noticed that the basal status is slightly up in many variables, and the effect of isoproterenol is slightly diminished, which is compatible with a higher level of phosphorylation in the basal condition, again compatible with reduced PP1 activity. Something like that. Okay.
Okay, thank you. So I will check.
Okay, any other questions? I just have a comment from Karen Cipido. She apologizes that she had to leave early. So,
okay.
Any other questions from anyone? Let's see, I got one from Raphael. Hi, Anna, great talk and beautiful data. I'm curious to know your thoughts about the Kuplan disarray and the junctophyllin alteration with the appearance of RYR substates. If I understand it right, is how is this structural change leads to the appearance or enhancement of subconductances?
Yeah, so I think that, so thank you, Rafael. We can just only hypothesize, so that it's someone who needs to do a structure that I don't know. Because the thing is in the slide that I try to find, so this mutation is in the middle of these three regions of the N-terminal that are held together by a chloride. So the arginine is negative and there are other residues that are negative. When this arginine is mutated to a neutral amino acid, like the one that we are analyzing, this binding to chloride is weaker. So that means that the stabilization of these three regions of the N-terminal regions, this is destabilized. So this destabilization may have that one of these parts get closer to the C-cord solenoid, and then maybe that it's why it interferes with the opening and closing of the channel through, I mean, even if it's far away from the opening, through this. So now, why this structural change affect junctophyllin binding? We still don't know where junctophyllin binds to the channel. So it's something that needs to be analyzed. But
yeah, it makes perfect sense.
To do it.
and really appreciate the impact of this finding. But an additional alternative could be that the stability of other sites are affected, like calcium binding sites, for instance, and that will explain the higher sensitivity to calcium and just the whole protein being dearranged, right?
Mm-hmm. Yeah. So that is this paper. The first author was Kim Lika, and the last is Philip Van Petegem, and it's in a structure. I forget the year. So if this mutation in the 420 is mutated to glutamine, it makes much more ultrastructural disarrangement that if it's mutated to a tryptophan, that it's also found in CPVT families. So this point mutation really makes a difference in the structure of the channel. Then it may, with junctophyllin, I don't know where it binds. It should not bind directly to the N-terminal because it's really in the middle, but it can have some effect on other regions, as you say.
Thank you very much. Nice seeing you.
Thank you.
Okay, I don't see any other. There is a comment from David Patterson who is saying, nice talk.
Okay, thank you.
And no question. And then
bye, Antonio
is leaving. And I will thank Anna again for a fantastic seminar. It was really a pleasure. And it's a beautiful study. Thank you so much.
Okay.
And before we leave, let me just remind everyone that 2 weeks from today, on September 28, we'll have Antonio Zaza from Milano, and he'll talk also about calcium dynamics related phenomenon, which is therapeutic circa enhancement as a target for therapeutic interventions. So Antonio, we'll hear from you next in 2 weeks. Thank you all for attending. Hope you had a good summer, and stay safe.
Okay. Thank you very much, Joram and everyone.
Yeah. Bye,
Nice
Anna.
to see you.
Hope to see you soon.
Bye.